Hello, my name is Arvind Kumar. I'm a faculty at the Royal Institute of Technology, Stockholm in Sweden. In this lecture, I would like to give you an idea of what a brain is, what makes it complex, and what is brain science. I will also give you a link to a series of videos that we have curated at the Neuromatch Academy. These videos are directed towards a newcomer to brain science. And with these videos, we would like to introduce basic terminology, give you an idea of main concepts and tools used in modern brain science. We hope that this will give you sufficient background to appreciate the complexity of the brain and to find your own trajectory in this fascinating world of brain science. This lecture, I would like to start with the dichotomy of the brain and the mind. Mind refers to the ideas and purposes. It's the ability of an agent to find invariances and regularities in their environment. Brain, on the other hand, is made up of material. It has its own chemistry and physiology. And the interacting components in the brain give rise to the dynamics. And this dynamics somehow implements what we call as the mind. And this is one of the most fundamental and complex questions, that how come the dynamics of this material object gives rise to computation, or something we can call as the mind. If you look at the brain, it's quite unremarkable in its weight, volume, and area, but it's very remarkable in terms of the energy it consumes. About 20% of body's oxygen is consumed in the brain. So brain has a very high energy budget. So it must be doing something important. When we think of the brain, we think of it as an organ. However, brain has distinct parts. For instance, here is a schematic of the neocortex of the brain. And in the neocortex, now we are able to identify specific regions which are dedicated to specific functions. For instance, in the occipital lobe, we have the so-called primary visual cortex, which is responsible for early processing of visual information. Similarly, we have auditory cortex, somatosensory cortex, the areas of Wernicke and Broca's. They are involved in language comprehension and production. Motor cortex is involved in motor control and execution. Frontal areas are involved in higher cognitive functions, and so on. Underneath the neocortex, we encounter the subcortical regions, which are evolutionarily the oldest structures of the brain. And they have also their specific functions. For instance, the hippocampus is involved in memory formation and spatial navigation. Amygdala is involved in fear processing and emotions, and so on and so forth. There are various components that can be identified which have distinct functions. But this itself is not sufficient that we can assign specific function to specific parts of the brain. To really understand how the dynamics of the brain gives rise to mind, we need to dig a bit deeper. And if we look at the cortical tissue, for instance, or any part of the brain tissue, what we will find is that it's predominantly made up of neurons. These are very specialized cells. This is one of the first drawings of the brain tissue made by Ramon y Cajal. And since his days, we have learned a lot about neurons. Neurons are very specialized morphologically. For instance, they have distinct structures. For example, dendrites, which are the input ports of this neuron. Then there's an axon, which is the output port of the neuron. When the conductance of the membrane is changed, a neuron responds with such a voltage deflection, which is called an action potential. And action potentials are the ways by which neurons communicate with each other. So these are very important, and essentially we can say that an action potential is the currency of information processing in the brain. Neurons, they come in a variety of shapes. As you can see here, these are the excitatory neurons, and in the bottom panel you see inhibitory neurons. I will introduce you what an excitatory and inhibitory neuron is in a minute. But if you inject some current in these, they also tend to speak very specific language. Each type has its own spike pattern, if you wish. There are a lot of neurons in human brain, about 86 billion. But this is not a remarkable number. There are animals and organisms which have smaller number of neurons. For instance, C. elegans has only 302 neurons. And elephant has more than 256 billion neurons. So just the sheer presence of large number of neurons doesn't make the brain complex. And neither the diversity of cell types is sufficient to make it complex. Coming to the diversity, you will hear more about the neuron diversity in the lecture by Christoph Koch. The second important component of the brain's hardware is the connections. These connections are called synapses. So when the brain first time comes into existence, this is when neurons are connected to each other. The connections among neurons are of two types, electrical and chemical. Electrical are really physical contacts, but more abundant connections are the so-called chemical synapses. And they are called chemical synapses because the axon of a neuron comes very close to the dendrite of another neuron. And that's where the connection is formed by release of a chemical. The presynaptic neuron releases the neurotransmitter, which is then taken up which binds to the receptors on the postsynaptic side, and this gives rise to a deflection in the membrane potential. Depending on the type of the neurotransmitter and the receptor, you can get a positive deflection in the membrane potential or a negative deflection. The positive deflection is called excitatory response, and the negative deflection is called inhibitory. Usually, in most of the neurons, they either express excitatory neurotransmitters or inhibitory neurotransmitters. And that makes a neuron either excitatory or inhibitory. And this is also called the Dale's dogma. Essentially, it gives us the first classification of neurons as excitatory and inhibitory. You will hear more about the neurotransmitter systems and how neurons talk to each other in the lecture by Emanuela Santini. Once we have the neurons and the connectors, the question arises how the brain is wired, how different components of the brain are wired. I'm going to take the example of the neocortex in here. So when we look at the tissue of the neocortex, first thing we realize is that it's organized as layers, 6 in total, counting from top to bottom. And within the layer, there is a very specific interlayer connectivity. Information arrives at layer 4, it is then transmitted to layer 2, 3, layer 2, 3 sends it back to layer 5 or 6. And both layer 2, 3 and 5, 6, these send their projections to other areas of the brain and receive input from them. So this is a motif which is repeated throughout the neocortex. Maybe not in the higher cortical areas because there the layer 4 is missing. If you look within a layer and look at horizontal connectivity, at the scale of about 1 millimeter, what we notice is that the neurons don't make arbitrary connections, but their connection probability decreases as a function of distance. This is true for different layers and also true for whether the neuron is making excitatory connections or inhibitory connections. And if we scale out a little bit, we encounter what is called the long-range patchy connections. This is a very nice drawing of tracer injection. So you inject some chemical in the neurons, which then travels through the axon, and you can observe it at the axon terminals. Here, a bunch of neurons were injected with the tracer, and you can see where they project to. These little patches are very interesting. There are specific rules, it seems, that are followed where the patch will be made. But for now, we can take it that short distance connectivity is distance dependent, but at longer distances, neurons make patchy connections. And if we zoom out, we can look at connectivity among various parts of the neocortex. Here it's a parcellation into 43 cortical regions. And one thing that comes out of this connectivity map, by the way, to read this map, these are the source regions and these are the target regions, and the color tells you the normalized connection densities. This doesn't tell you the strength. But what pops out of this figure is that first of all, the connectivity is definitely not random. It seems to have some structure. In fact, we can assign some components as specific modules, and there seems to be some sort of hierarchy in this. And this is where I think the brain's complexity arises. This particular type of inter-region connectivity is largely determined by the genetic makeup. It is species specific, and maybe there are even individual specific differences. So this is where we first time start to appreciate what makes the brain complex. It's not the number of neurons, it's actually the connections. In the brain, on average, a neuron sends out about 10,000 projections and receives 10,000 inputs. This is very remarkable. We don't know any other system which has such large amount of interactions with other components. Not only that, the connections are made by traveling activity along the axons, and the conduction speed is decent. It's not very high. It's about 220 miles per hour. But this means that areas which are far away, there will be certain delay. So this connectivity is dense in the sense of how many connections a neuron receives, but it's sparse in terms of given the large number of neurons. But still, the connections have some delay associated with them. Moreover, their strength keeps changing because of learning, because of development, and so on and so forth. So that's what I think makes the brain complex, that its connections are delayed, there is a dynamic change in the connection strength, and there is some structure to the connectivity. What comes out of this is that we can try to approximate the brain as a network of networks where there are local regions which have their own connectivity rules, and then there's a new set of connectivity rule which connects the different regions. And it's the interaction of the local activity with the interregional activity that gives rise to the function. With this, I think we can come back to, like, once we have this complex system, we can ask, how do we approach the brain function problem? Well, the idea is that we would first like to relate the behavior, the output of the brain, to its activity. And that's what we would call as the brain function. To relate the behavior to brain activity should give us some glimpse of what the brain function is like. And to study this, the approach is quite intuitive. Animals are trained to perform specific tasks, and we do record brain activity in specific brain regions using various methods. Simultaneously, we also track their behavior, and by relating the behavior to the neural activity, we get some clues into the organization of this particular function that is in the experiment. So this kind of gives us the neural correlates of behavior. But of course, correlation is not sufficient. We would like to find some causal links between the brain activity organization and the behavior. And for that, we need to do perturbation experiments. These perturbation experiments come in various forms. Sometimes these are just lesions or brain diseases. In other cases, we can create a controlled reversible lesion in specific parts of the brain, in fact, even in specific cell types. And that gives a very good experimental paradigm to uncover the causal links between brain activity and brain function. In addition, we also try to find analogies between brain function and the most advanced technology around us. For instance, in modern times, we try to relate the brain function or its activity to modern machine learning algorithms. And therefore, a number of experiments have been designed to test whether a certain machine learning algorithm could be implemented in the brain. And reinforcement learning is a great example of this. When it comes to reading out the brain activity, it's very interesting that we can read out the brain activity at various spatial and temporal scales. There's a constant discussion about what is the right readout of the brain. Sometimes it's a matter of convenience. Sometimes there are good reasons to record one or the other form of brain's activity. You can read out brain activity in the form of individual neurons spiking, and that gives you a very fine spatial and temporal resolution. If you're reading out things like electroencephalogram or magnetoencephalogram, there we are reading out activity of a bunch of neurons together. So we can't have a good spatial resolution, but temporal resolution is maintained. Things like fMRI, which are really an indirect measure of brain's activity, because here we are really reading out the blood oxygenation. So they give you a relatively coarse spatial and temporal resolution. Spatial resolution, of course, depends on the choice of magnitude, and so on and so forth. So we have a variety of tools to record brain's activity in different resolution. And that's where we have created a series of lectures at Neuromatch Academy to give you an introduction to variety of brain's readout, from spiking activity to calcium imaging, local field potential, EEG and MEG, and finally even fMRI. Once you have the activity, the question arises what kind of behavior we are going to study. Study of behavior and measurement of this behavior is covered under this lecture by Professor Reed on psychophysics. You will also see a very nice video from the lab of Professor Anne Churchland, who kindly recorded ongoing work in her lab and gives us an idea how this is done in real. You will also learn about what kind of behavioral readouts we can measure automatically and relate them to neural activity. And we have a lecture on how information is represented in the brain. Jens will focus mainly on the spiking activity readout of the brain. And finally, we hope that we can wrap it up, and Christoph Koch will give us a glimpse of how we can study the brain as a whole and go from neurons to the big problem of all, for instance, the consciousness issue. So that's our Neuromatch video series, which hopefully gives you an idea of how the brain science works, right from the planning of experiments to recording of brain activity and asking good scientific questions and even addressing the problem of consciousness. Thank you very much.